You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have David Pamies. He's on the Faculty of Biology and Medicine, University of Lausanne in Switzerland, used to be at Johns Hopkins. And we're going to be talking about a human-induced pluripotent stem cell-derived 3D platform. So David, thank you for coming.
No, thank you for having me. It's a pleasure to.
Yeah, for people that don't know, what are induced pluripotent stem cells?
So induced pluripotent stem cells is basically when you have adult cell, and then you can reprogram the cells to become, again, stem cells. So that allows you to get, stem cells from different patients without going through the, taking the cells from the embryos. That was a big ethical problem before. So you can introduce certain factors inside the somatic cells, and they become, again, stem cells. So that's basically induced pluripotent stem.
Right. Which, what cells are you trying to recreate in your particular experiments?
So what we are trying to develop is in vitro models that represent human brain. So basically what we try to differentiate the cells we're trying to do it in a 3D structure. So basically we generate like a small tissue that has different neuronal cell types, astrocytes and oligodendrocytes, the different types of cells that we will find in the brain. So we differentiate them all together so they are interconnected and they resemble a little bit or mimic a little bit what we will find in brain tissue.
How, the brain is like incredibly complicated. So how much of the brain's function are you trying to approximate with your spheroid?
Yeah, that's a good question. So in our case, what we are trying to do is the application we would use is for toxicology. So we need to find a way that we can find something that is complex enough to kind of reproduce some functionality of the brain, but that is not super complex, because when you get like a really complex models, then it's really difficult to have a reproducible system. So in toxicology, we want to have a model that every time we produce this tissue is the same. So then when we introduce chemicals and toxicants or drugs, we don't have the variability of the model. So in our case, what we have is, let's say, the basic tissue unit. So we have, the neurons that are connected between them and they have a spontaneous electrical activity. We have the oligodendrocytes wrapping with the myelin, the axons, and astrocytes supporting neurons, stuff like that. But what we don't have is like the different region of the brain. There's obviously some parts of the brain that have a specific type of neurons or things like that. So we have something that is kind of general, so that they reproduce a little bit what will be the general structure of the brain, but it doesn't have the particularity of each different region in the brain.
Yeah, I'm sure that'll be nearly impossible to recreate. Are you assuming in terms of toxicology that the substances you're testing have made it past the blood-brain barrier?
Yes. So we also have incorporate in our model a blood-brain barrier system, so we can check if the chemicals will pass the blood-brain barrier or not before testing in our system. But also there's by chemical properties of the drugs, sometimes you already know if this is something that is going to happen most likely or not. But you can also test this in vitro to check if the chemicals will cross the blood-brain barrier or not.
And where do you get these cells from? Do they start out as skin cells, or what do they start out as before you bring them back to pluripotency?
So normally we get them from donors. We take from the skin, and then from the skin we reprogram them to become stem cells. And at the moment we have several lines that are coming from healthy donors, but also from some disease. So we have, for example, for Parkinson's disease, we have some autistic lines from autism, things like that. When we get the glioblastoma, because we also use the model for drug screening in glioblastoma research, then the glioblastoma are obtained directly from the patient. So when the patients die, then they remove the glioblastoma, and then we incorporate these cells into these brain spheres.
So have you seen that certain conditions do translate to the cells themselves? They're preserved even through pluripotency, and certain ones are not?
From my experience, I haven't seen much differences. We have some. There's some studies showing the differences between a disease. In my hands, I haven't seen that, but there's some studies showing, like, for example, some schizophrenia patients has a lower myelination in the axons and things like that. However, sometimes it's really difficult to know if it's because of the disease or because of the reprogramming. So you have to be really sure that the cells, it could be also by the variability between individuals, and not because of the disease. So you need to have several lines from patients. You have to have several lines from healthy patients, and then you have to derive in the same way, because there are different ways to reprogram the cells and differentiate them. So you have to make a lot of controls and have a lot of lines to make sure that the differences are because of the disease and not because of the variation of the process. But there are some interesting research already published showing differences in some disease.
When does brain toxicity tend to occur when someone takes a drug? Is it very quick, or does it happen later on after the drug has been metabolized by the liver and other organs?
Well, what we are doing is a little bit different. What we are trying to do is to predict the toxicity that will happen in humans. So at the moment, the current guidelines are in rodent mainly. So people use rats or mice to do the different tests to see if a chemical is teratogenic or produce eye irritation or things like that. So what we're trying to develop is a test that is in human-based test, and also in vitro, so it's faster and cheaper, because at the moment the problem is that we have so many chemicals we don't know what they are the toxic effects, and the guidelines are super slow and super expensive, so it's really difficult to test all the chemicals. So we're trying to find something that is really fast in order to predict these chemicals. So what we are doing basically is we are not in the pharmacokinetics, so we are not checking how the drugs are going through the body and so on. We are more trying to identify which chemicals could produce some effects in the brain, and chemicals that we expose in the environment. For example, the flame retardants that we have in the tables to avoid that they start to a fire. And these kind of chemicals we are exposed all the time, and we don't know what effects they have in our brain. So where we are trying to develop tests that are cheap and fast enough to be able to test many chemicals and better mimic human physiology and able to predict better than the animals, because at the moment animals have a really bad prediction also in humans. And recently we have also tried to study also in the drug development field, but mostly focus on toxicology, also how different drugs will affect glioblastoma tumors, but also kind of in the same way. So we are now working in how the chemical goes through the body and is metabolized. Is mostly to see which drugs are more efficient to kill the glioblastoma tumors without killing the healthy cells. So this is more or less what
but the issue may be, what if you test all these drugs and they don't look like they have toxicity, but in vivo, let's say they're metabolized by the liver and processed by different parts of the body, and then they're toxic. You wouldn't know that without hooking up, a bunch of other organoids from different parts of the body. So
you are completely right about
this.
So what people are doing now is they are trying to develop in silico models. So they are trying to do some studies, direct studies into how the drugs are metabolized and so on, and then try to model by in silico. So they do in computer science, they try to model a model that could predict how the chemicals are going to be distributed in the body, how they are going to be metabolized and so on. So then they try to compare this in vivo and try to see if the models works and things like that. And also there are some people trying now to link this with chemical structure. So they can predict, for example, if you know that these 10 chemicals have a similar chemical structure and they will be metabolized in a certain way or distributed in the body in a certain way, then how much you can predict a chemical that also have a similar chemical structure. And that's how people are trying to do this. However, until probably you go to if it's in terms of a drug, until you go to clinical trials and things like that, it's really hard to know like 100% what is going to happen with the drug. But what you want to at least try to have the safest drug going to clinical trials. And it's a big problem at the moment because it's the same way that with the animals that, so if you check the statistics, how many drugs that are developed going to the market, only like 10% of the drugs developed going to the market, and mostly they fail in the clinical trials. And this is because the main model that we have at the moment, also with the in vitro system, no, you are trying to develop something in a rat model that obviously is not a human, and then you use different concentrations per kilogram and things like that, and then when you move to a human, then most of the problems with the drugs is related with efficacy or with safety. So the drugs doesn't behave as you were expecting, and also they are more toxic than they were expected in humans. And that's why all the drugs fail. So there's a big issue at the moment to try to figure out how we can solve this problem and how we can make drugs candidates more efficient, the selection of the candidates more efficient. So we don't waste so much time because clinical trials are really expensive. So you have to be 100% sure that you are going to start clinical trials because people don't have the money to do many of those, right?
But if clinical trials are so expensive, why don't the various organoid creators get together and cross-license their organoids so that they can, get a much better approximation of what really goes on? Or like, let's say you're going to test, 500 compounds this year. If you don't create the liver organoids yourself, why not partner with a company that does and send them all the chemicals and partner with them on the results? Hey, when you guys run it through your liver chip, are you getting the same thing out as we're getting?
Yeah, I totally agree with you. I think there's people trying to connect different organoids together into that's what a few years ago was this huge project in US that was funded by FDA and DARPA and NIH, trying to do the human on a chip, so they will have all these organoids together into the chip. And there's some publications showing the connection of a few organoids, like for example, liver, kidney. But the problem is to connect all the organoids at the moment is not so easy because you need to solve the problem of having the same media for all the cells. Also have kind of similar stages of differentiation in all the cells because, cells might be in the different stages There's even some cells that will never be able to completely fuel, maturate. So it's not so easy to do this. But I agree that if people will share more the information, it could be easier, to reach to a good point. However, pharma don't want to normally share any information about anything that could be profit from them. Inside, it's already hard in like public science because, publications is putting all the pressure. So you want to be the first and things like that. But if you move to the pharma companies, it's even worse because, they know that if they have a really good candidate, it could be a lot of money. So if you want to know, how this is metabolized in another organoid, but you don't have it in-house, you have to share the information with someone else and this type of things. It gets complicated. And yeah, also, I think pharma is, as all these organoids and organ-on-a-chip things are really new, I think pharma is still like a little bit like seeing what happened and before going into this type of research. And we know because we have some contact with some pharma industry and here in Switzerland, they are really interested, but they obviously don't have yet in-house any of those systems, right? So they are talking with us to see how they can use the models and how they can incorporate this to the pipeline or the initial drug screening and things like that. But as it is quite new, I think pharma is still a little bit not really into the game yet.
Who first came up with the idea of an organoid and when? Do you know?
Well, 3D models so it depends what you consider organoid, because also the terminology is changing in the last years, but it has been already known for a long time, 3D cultures and 3D cultures that already resemble a little bit the functional part of an organ. And this has been really from really long time. In the case of the brain, in 2013, there was Lancaster paper that was, I think, was a Nature paper, and they were the first time to show that they can differentiate the brain in with the different with a little bit of the structure of the initial development of the brain. So they have different compartments of the brain with different sections. You could distinguish between forebrain, midbrain, and this type of things. And this was in 2013. But there's other organs, like liver organoids and things like that might be a little bit earlier than that. But I would say maybe, for the new organoids, I would say maybe 14 years or maybe 10 years. But there has been already people working on, rat organoids, rat 3D models from really, really long time ago.
In making the organoid, you're just making like a spheroid. I mean, are you seeing that the cells are moving around and rearranging and creating their own morphologies as you differentiate them, or like what do they do? Is there any interesting behavior there?
Yeah. There's definitely some. They are mimicking a little bit the process that we could find in the development of the brain. For example, you could see how the different cell populations will differentiate in different stages, right? So, for example, you will see first the differentiation of neurons and astrocytes, and then the oligodendrocytes are coming later on, and then the oligodendrocytes, after a while, they're starting producing the myelin that will wrap the axons. These type of things, you can see them. Also, you can see, for example, a high proliferation of oligodendrocytes in the beginning of they start to differentiate, and then how they are, they are selecting only the oligodendrocytes that are functional, and the other ones are dying over the differentiation process. And this is something that also happens into the normal development of the brain. So you can see these type of things. It depends how complex is the model. If you go for like cerebral organoids, you can see also how the stem cells migrate and then differentiate over the migration, and the different stages of differentiation through the migration, and these type of things. I can only talk about brain because it's my field, but I'm pretty sure you can see also in other organoids how is mimicking kind of the differentiation that happen in the normal in vivo situation.
Are you using a scaffolding, or how do you make sure that the the organoid forms in some kind of sensible way?
In our case, we are using a bioreactor. So basically the cells are floating and it has a certain movement to avoid that they grow in size. Because in our case, we want to keep them smaller than 400 microns, because when for example, the cerebral organoids that we were talking that was published by Lancaster, they grow really big and they have different size. However, they starting getting necrotic centers in the middle. And that in toxicology or drug screening is not possible to use because then you don't know if the toxicant that you are putting is the one causing these necrotic centers or not. So in our case, we are trying to keep these neurospheres or brain spheres smaller than 400 microns so they don't get necrotic centers. So we use this spinning movement to keep them really homogeneous in size and in shape. So that's how we culture the model.
So the organoid forms into like a spheroid shape of what kind of typical dimension?
They grow around, it's like a sphere and it's around 350 microns. When we introduce the tumors, so like I say, sometimes we use this for drug screening for a glioblastoma. What we do is incorporate a few cells of the tumor inside the sphere. And then over the weeks, the tumor is growing and start interacting with the cells in the surrounding. And then in these cases, sometimes you can see that the brain spheres with the tumor grow bigger than 350 microns. But it's because of the growing of the tumor.
Interesting. All right, so you're modeling these tumors as well, and then testing drugs on the organoid with the tumor in it.
Yes, so something that we have, I think we are the first in doing this. Normally in glioblastoma tumor field, the people has been using the tumor derived cells from patients and then grow them in 3D. However, they are growing normally only the tumor cells, and they have been testing different chemicals in these tumor cells. However, we know that the tumors, especially glioblastoma, interact with the cells in the surrounding, and there's a crosstalk between the healthy cells and the tumor for the progression and the survival of the tumor. So I think where we wanted to be sure that we have the tumor into a microenvironment of the tumor, so we can also see the interactions between the tumor and the cells in the surround. But also this is really interesting when we do the drug screening, because normally drug screening is based on the tumor, right? So you will see the effects of the drugs into the tumor, but you don't know if the drug is going to also affect the healthy tissue, because some of the chemotherapies also are, some toxic, they have some toxic effects in the healthy brain. So with this system, you can check the tumor, if the tumor is affected by the drug, but also if the healthy cells are affected by the drug. And we have incorporated this in a high throughput platform, so we can measure hundreds of drugs at the same time. And now we're trying to get more funding to do a bigger test with more than 100 chemicals into drugs into the glioblastoma that we have.
How does the tumor act versus the regular cells? Do you see differences in behavior, and so what do you notice about them that's different?
Yeah, definitely. First the tumor is growing faster, and normally when we have the brain spheres, when cells differentiate, normally they don't proliferate a lot. For example, neurons will not proliferate after they are mature. So in some point, the brain spheres stop growing, and it's just maturating. So they are increasing the connections between the cells and stuff like that. However, the tumor is still growing, and you could see some markers that are, for example, for proliferation that are really present in the tumor, while in the healthy tissue you don't see it. And what we are trying to do now is we are going to try to study what are the molecules that the tumor is secreting to the healthy cells, and how the tumor is changing the healthy cells around the tumor. That's what we are trying to focus now. But definitely, they have a completely different behavior, the tumor with the healthy cells.
Yeah, is there any immune system that normally would be there in the brain, and are you able to mimic that in the organoid at all, or is that absent?
No, actually, immune cells are really important for development, for glioblastoma development. And in fact, microglia, that is the main immune cell in the brain, are really interacting with the tumor, and the microglia migrate into the tumor, and they are the main key players in the model. Because of the microglia are derived from different cells, not from the initial source of what we use, and we use neuroprogenitors to make these brain spheres. The microglia are derived from different lineage. So what we are doing now is, in collaboration with Sally Cowley in Oxford University, we are deriving microglia from iPSCs, and we are incorporating also the microglia into the sphere. So we have the brain spheres with the microglia, and we already have this in-house, and we are doing different, we are trying to activate the microglia with different stimulus, with inflammation, cytokines, and things like that. And what we are trying to do now is also to incorporate the tumor and see how different, for example, we are trying different drugs, like, for example, doxorubicin or the temozolomide, and we want to see if the sensitivity of the drugs change when we have the microglia into our model. So that's something that we are working on because we know that microglia is really important for the glioblastoma development. So we are trying now to solve that issue, and hopefully we will get some interesting results in the next year.
This is a weird question, but could you ever grow an organoid just from cancer cells? Do they differentiate and form any structures, or they just seem to just go on a dividing rampage and they don't bother to differentiate or anything?
Actually, when in the glioblastoma field, when they take a glioblastoma from a patient and they do some cultures derived from this glioblastoma, in many cases they call them organoids already to these models in 3D, because glioblastoma is also it's not really homogeneous, so have different cell types into the glioblastoma, and also you can see certain kind of structure, and so it is not like the glioblastoma is simulating any like organ, but they have their own structure into the glioblastoma tumor per se. So they start getting necrotic centers, and that will help to secrete some growth factors that will bring the vasculature and things like that. And you can see how cells are distributed differently into the tumor. So there are many people already call them organoids to these tumors per se, because they have a kind of a specific structure.
I just wonder, I mean, could you make, so you literally could have a brain organoid that is entirely cancer cells, and would it look anything like an organoid of healthy cells?
Exactly, yeah. You could have, so basically the word organoid, from my opinion, it means that they're simulating something that is happening in vivo. So you can have an organoid that is simulating what is the brain, and you can have an organoid that's simulating the tumor, right? So they are completely different. One is simulating trying to represent like a healthy tissue, and the other one is trying to represent more like a tumoral tissue. But it's a problem with the definition at the moment, because people call some things organoids, and other people don't consider this organoid. Other people say that they are organotypic cultures and microphysiological systems. And at the moment, it's a little bit weird, all this definition. And also because people in cancer has been calling these organoids for a long time, and now we use the word for other things, and it's kind of confusing.
It makes sense. So any really interesting things that you've discovered recently from your work on these organoids that you can talk about? Anything that jumps out at you that was really surprising?
Well, I think the most interesting thing that we have been, like I say, we are more into the testing field. So something that we has been studying lately has been antidepressive drug that pregnant women normally could take it because of the pregnant depression. And we have been studying the effects of this drug in the brain development. And what we have is developed an assay, a test that has different endpoints that measure different key events during the brain development, like, for example, neural aggro, synaptogenesis, myelin formation, oligodendrocyte maturation, so several things into the same model. And we have tried to use, so we have tested the drug in with the blood concentration that pregnant women will have, and we have tested in our system, and we have been able to see some effects in the brain development into our test, indicating that this drug might have some effects in the development of the brain when the pregnant women are taking. And that could be, really interesting to see. If it's the case, then we should put the alarm, in the government, so we can restrict the use of these drugs.
Yeah. So some of the organoids are they made from male lineages or female lineages, and is there any difference when you culture them?
So we have several lines. We have from male and from female. We haven't seen differences in terms of differentiation. At least the lines that we have. We haven't done any study comparing female and female at the moment, male and female at the moment, but we don't see differences in terms of differentiation. But there might be some differences in terms of transcriptomics or some proteins might be different. But as we don't have the hormones into the differentiation process, and hormones might be really important and to see the differences between sex, between female and male. We haven't seen yet anything at the moment. So, but I don't know because we haven't worked in that field a lot. But I know that many people now are trying to see the differences between men and women lines. There's obviously differences between individuals. So there's some polymorphism that might make some lines more sensitive to certain chemicals than others. That's why it's important that to study not only one line. And when you test a chemical, you want to have several lines, so you at least have a little bit of heterogeneity in terms of polymorphisms and things like that.
And one thing I haven't asked, I forgot, is how do the organoids get food, nutrients and oxygen? How do they respirate and how do they get food?
Well, so when they are like in the media, and the media has already the nutrients that they need to, they basically absorb the nutrients from the media. So it will be like extracellular, the extracellular matrix will provide the nutrients. The oxygen also, as we don't have vasculature, and at the moment there is no much advance in trying to put vasculature in the organoids, they also have to get the oxygen through the, basically taken from the media without any vasculature that brings the oxygen. And that's why, when you get a really big organoids, you start seeing how the cells in the middle are dying because the oxygen cannot fuse into the centers of these organoids. And I know there is already some publication showing that they are trying to generate also a vasculature into the organoids, so then the oxygen will be able to reach all cells into the organoids. But at the moment, you can only see like there is no complete the vessels, and it's not completely functional yet. But I know there are some people trying to work on this to be able to reach the oxygen in all the cells. Also, many people use like bioreactors or shakings, so the cells are in movement, and they can you can have a higher interchange of oxygen between the media and the incubators. But that's only limited. So you cannot get really big tissue without vasculature because, not all the cells will be able to get the nutrients and the oxygen.
Okay, got it. Well, very good. So what's the best way for people to find out more about your work and maybe contact you and interact?
Well, there is. If they want to contact me, they can look for me in the website of the University of the Department of Physiology in Lausanne University. There's also, if there's scientists interested, they can look for my publications, and I'm the corresponding author in most of the last publications, so my email is there, so they can contact me. And yep.
Okay. That's great. Well, David, thank you for coming. It's been a very good call.
Thank you very much for having me.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.